workshop. And this time I know, is very hard for everyone because of the COVID-19 pandemic. So I hope you are doing well, staying safe and healthy. Besides the COVID-19, the tick-borne disease also have the serious impact on the animals and humans' health that many scientists and researchers are focusing on, because the tick can carry various pathogens, including the viruses, the bacteria, and the protozoa that can infect animals and humans. Therefore, the tick-borne disease are also considered as a global and health concern. In the Southeast Asia, more than 90 tick species have been recorded. However, very little is known about their ecology and also the population dynamic. The information about the tick-borne disease is also limited in this area. So being supported by the Academy of the Medical Sciences Global Challenge Research Fund Networking Grant, connect one can update a lot of information through today's workshop. So to kick off this workshop today, it's my honor to introduce to you Dr. Bhui Anh Dao, the Dean of Faculty of Veterinary Medicine, Vietnam National University of Agriculture, for opening remarks. So please, Dr. Dao. Dr. Dao, are you there?
Yes, I'm here.
Okay, so please, Dr. Dao.
I have a problem with my webcam.
So it's okay, please send us the message.
Yes. Good afternoon, ladies and gentlemen. It is my great pleasure to welcome every one of you to the 2021 International Symposium on Tick and Tick-Borne Diseases, a One Health perspective on the project strengthening research capacity on tick and tick-borne diseases in Southeast Asia, under the collaboration between the University of Bristol from the United Kingdom and the Vietnam National University of Agriculture. The current project is sponsored by Global Championships Research Fund Networking Grant. The funding has provided us the golden opportunity to be in a collaboration with Professor Richard Wall from the University of Bristol of United Kingdom and Dr. Tawin Ipanki from Kazakhstan University, Thailand, and Dr. Malaika Watanabe from University Future, Malaysia, and many other researchers from all over the world, expertising in this specific subject, ticks and tick-borne diseases. This project has brought together a network of researchers from Southeast Asia and the wider region, along with researchers from United Kingdom, to discuss best practice approaches to tick control. The network would further build capacity in the local expertise able to address to complex issues associated with the tick management. We are overwhelmed and grateful to have your presence, and hoping that you enjoy the quality of the research present at the symposium. I wish you continue a good health and success. Thank you.
Thank you, Dr. Dao, for your welcome speech. And in our workshop today, we will have the two presentation sections. The first section is about the tick and tick control. The second section is about the tick-borne disease in animals and humans in the Southeast Asia. So after each presentation section, we will have a short Q&A discussion. So please do not hesitate to leave any comments and questions in the chat box on the Zoom platform or comments on the Facebook platform. So right now, please allow me to give you the brief introduction about 7 key speakers today. So the first speaker is Dr. Bryony Sen from the University of Bristol, United Kingdom. And her talk is about the climate and the seasonal abundance of the tick Dermacenter reticulatus. The second speaker is Dr. Katie Liho from the University of Bristol, United Kingdom, with the predicting current and the future risk of the ticks on the UK livestock farm. The third speaker is the Professor Richard Wall, and he gonna give us some information about using the essential oils for the prevention of tick attachment to humans and companion animals. And after that, we are moving to the second presentation section, start with the Dr. Tawin Ipankep from the Kansas University, Thailand. And we also have the Dr. Malaika Watanabe from the University Putra, Malaysia. And next speaker is Dr. Bhui Heinlein from the Vietnam National University of Agriculture. And she will update us some information about the tick-borne disease situation in animals in Vietnam. And the last speaker today is Dr. Nguyễn Văn Dũng from the Nimpa, Vietnam. And his talk is about the situation of ticks and tick-borne disease in humans in Vietnam. So thank you so much all speakers for being here today. And I think right now we can start. And the first speaker is Dr. Bryony. So Dr. Bryony, take your time. Dr. Bryony, are you there?
Yes, it's saying I can't start my video. Is that, shall I just share my screen? Yeah.
Can you share the screen right now?
Yeah, that's fine.
Okay, so please take your time.
Okay, hi, yeah, I'm Dr. Bryony Sands from the University of Bristol, and I'm going to talk to you about climate and the seasonal abundance of the tick Dermacenter reticulatus. So this is a 3-host exodia tick that's widespread in Europe. It's recently undergone range expansion throughout Europe, for example, northwards through Germany in the past 50 years. Despite its widespread ecology, despite being widespread, its ecology is not well understood, and that's largely due to the elusive nature of the immature stages. So they're thought to be nidicolous, which means they remain in the burrows of their small mammal hosts, and they're very rarely collected. They can be collected by trapping small mammals, but not really by blanket dragging. So they're an important disease vector, and it's important to get a deeper understanding of their biology and epidemiology because of this. And they transmit a number of disease organisms, including Barbasia canis, which is a fatal pyroplasm of dogs. And that was first found in untraveled dogs in Essex in the UK in 2016. And it was confirmed to be carried by a population of D. reticulatus in the area. So there's 4 known regions in the UK where D. reticulatus occurs. West Wales, which is a coastal dynamic sand dune area. North Devon, which is also coastal sand dunes and grazing marsh areas. South Devon, which is maritime grassland, and Essex, which is grazed coastal grassland and marsh, as well as a population in Harlow, which is an urban parkland inland. So these small isolated populations have been known in the UK for at least 100 years. But you can see this is a map of Europe here, and there's the UK. So you can see that it's at the edge of its geographic range here. And it's interesting to note that it appears to be restricted to coastal regions here in the UK. However, in mainland Europe, that's clearly not the case. It's easily identifiable due to this ornate scutum, so it's the only tick in the UK with this characteristic, except very rarely D. marginatus, but that's only really found in Europe. So the aims of this study were to quantify the seasonal abundance of D. reticulatus in the field, to investigate associations with climate and habitat, and to quantify the effects of temperature and relative humidity on survival in the laboratory. So for this seasonal abundance work, we performed blanket dragging to catch the questing adults, and we also did burrow swabbing to try and catch the immature stages, and I'll talk about that in a minute. So we went to 6 sites, 2 in West Wales, 2 in North Devon, 1 in South Devon and 1 in Essex. And these were sand dune, marsh and maritime grassland habitats, all of them with grazing animals, including cattle, sheep and horses. And they were dog walking areas characterised by rabbit warrens. So the rabbit might be an important intermediate host here. So to achieve randomized surveys of the sites, we generated 200 by 200 meter grid maps and randomly selected 5 coordinates for each visit. And then we did 5 25 meter drags at each coordinate. So that was 625 meters sampled per site visit. We used a 1 meter by 1 meter squared white cloth blanket. And you can just see some of the D. reticulatus that we collected there. So this is my attempt at showing you how we did the burrow swabbing. It was basically a flexible tube that we put down the burrow, covered in cloth until it got to a dead end. However, we only found 4 immature ticks over the whole 12 months. So either this swabbing wasn't effective, we did catch fleas, rabbit fleas and mites on that, or the immature stages don't spend much time questing in the burrows, but we just don't really know at this stage. So we did 2 to 3 sampling trips per week. So with the 6 locations, that means that most of them were sampled every other week for the whole 12 months. And we recorded vegetation for each drag, and all of the adults and juvenile ticks were taken back to the lab. So in the lab, we looked at the temperature and humidity constraints, and we used these glass desiccator jars with potassium hydroxide solutions to create specific humidities inside the jars. So we had 5 humidities here, and then put them in incubators at 3 different temperatures, 4, 15 and 30 degrees C. So we had 15 desiccator jars in total, and we had 3 tubes each with 5 ticks. So 3 repeats of 5 ticks in each jar. So we used 225 ticks in total. And we continued the experiment until all the ticks were dead or until 9 weeks had passed, checking for mortality every 2 to 3 days. We also examined freeze tolerance. So we put the ticks in at -4 degrees C for each of the 5 humidities. And we also looked at cold shocks. So extremely low temperatures here down to -20 just for 24 hours. Looking at the results, so this is the field collected ticks. This is a graph of the seasonal abundance. So we have ticks per 500 meters squared, and then the months of the year along the bottom. You can see that there were no questing ticks between July to October, and there were peaks in activity around October to November and February to April. So we also calculated saturation deficit using temperature and humidity data from local field sites, and we've just plotted that in that red dotted line there. So there was a significant negative correlation between saturation deficit and tick abundance. This is the same graph here, except that temperature is plotted on there in blue instead. And that's just to show you that there were no questing ticks found when the 5-day average temperature exceeded 15 degrees C. Looking at associations with vegetation, there was a significant association with creeping willow, as opposed to the other vegetation types, which included bramble, marram grass, meadowgrass, rush and woodland. And this is a picture of the creeping willow here. So you can see it's very low lying and dense, and possibly that creates a better microclimate for maintaining saturation deficit. So these are the results of the lab work. So this is tick survival, the number that were alive, and humidity along the bottom, and then each line represents different temperatures. So there was a significant relationship between humidity and tick survival for all 3 temperatures. You can see that more ticks survived in colder temperatures. So this one's 4 degrees here, 15 and 30, and few ticks survived at low humidity. So they need cool, humid conditions. We also calculated saturation deficit using the temperature and humidity data from the lab work, and we found that there was a saturation deficit threshold of 0.7 kilopascals. And that agreed very well with the field study. So going back to this graph of the seasonal abundance here with saturation deficit plotted, you can see that here at about around 0.7 kilopascals, there were no questing ticks collected. This is the freeze tolerance. So this is tick survival at -4 degrees for 13 weeks. This is not a significant relationship. So there's much less of an impact of humidity on survival at -4 degrees. And around 20 to 50% of the ticks could survive -4 degrees C for 13 weeks. And then finally, the cold shock. So these were -9 down to -20 degrees for 24 hours. You can see that they could survive down to -18. They're about 20% surviving, about 50% surviving at -15. So that's quite an impressive ability to withstand cold shock. So in conclusion, this tick is known as the winter tick, and we confirmed winter activity. However, we did find peaks in activity in autumn, October to November, and early spring, February to April. So it can survive long periods below freezing and extreme cold shock for short periods. So possibly in those very coldest months in the middle of winter, sort of December, January, it's out there but not questing, and then it begins to quest either side as the temperatures rise slightly. So it's likely to be more cold tolerant than other species like Ixodes ricinus. It would be useful to do direct comparisons between these species. But we do know that the lower lethal temperature for Ixodes ricinus is -10 to -5 degrees, whereas here we've shown that for Dermacentor reticulatus, the lower lethal temperature is between -20 to -18. So it does seem to be more cold tolerant. We found a strong association with saturation deficit and an activity threshold of 0.7 kilopascals that was confirmed both in the lab and the field studies. So in the UK, the saturation deficit's only really above 0.7 kilopascals for short periods in the summer. So it's likely that temperature might be a more important factor in this relationship in the UK. For example, we did see that above 15 degrees Celsius, we didn't find any questing ticks. So in terms of climate change, it might be less likely to be active in warmer summers. However, because it easily survives for these freezing winters and appears to begin questing as the temperatures sort of rise above zero, the less severe winters that we see with climate change could explain the range expansion to more northerly latitudes and enable more questing in milder winters. So the spatial distribution in the UK still remains something of an enigma. It is at the edge of its current geographical range here, but there appears to be no climatic limitations to its spread. And we have seen that recent inland population in Harlow in Essex. But any range expansion in the UK would bring with it an elevated risk of disease transmission, for example, canine babesiosis. So what's next for this study? We're currently doing PCR analysis of DNA from the ticks that we collected in the field. This is in progress. We've done about half of them so far. And we've been looking for Babesia, Anaplasma, and Borrelia. So far, we have found Babesia associated with some different animals. So that's cattle, horse, dog, deer, and rodent. And these are animals that were found at the sites. For example, there were grazing cows and horses there. But yeah, that's still in progress. We're also going to be modelling the UK distribution with future climate predictions. So that's it. Just going to thank you for listening and thank the Dogs Trust for funding the study, Richard Wall and Katie Breyer for participating in the work, and Kaylee Hansford and Liz McGinley from Public Health England who sampled the Essex site for us.
Thank you so much, Dr. Brownie, for your sharing. And I think we are gonna return back to you at the discussion part. And
Okay.
right now we have to move to the second speaker, Dr. Katie. Dr. Katie, are you there?
Hi.
Hi, Dr. Katie. Could you please share the screen? Please take your time.
Okay, so hi, I'm Katie. I'm a PhD student at the University of Bristol. And today I'm going to be talking to you about a model I built to predict the current and future risk of ticks and tick-borne disease on livestock farms in Britain. So the main tick species in the UK is Ixodes ricinus. And this diagram shows a simplification of its life cycle. It's a 3-host tick, so it has 3 on-host blood-feeding stages, each separated by an environmental stage and a molt. And it finds its host by the process of questing, which you can see in this picture on the right. And as it progresses through its life cycle, it tends to quest higher in the vegetation, and this loosely correlates with the size of host that it feeds on. So as well as livestock hosts, Ixodes ricinus also feeds on wildlife hosts. And a particularly important wildlife host in the UK is deer, because these feed all 3 life cycle stages, including the reproductive stage, adult females. In the UK, Ixodes ricinus transmits several different diseases to livestock. And these are redwater, caused by Barbasia, tick-borne fever, caused by anaplasma, louping ill, and tick pyemia, which is caused by the bacteria Staphylococcus infecting a tick bite. There's no vaccines licensed for use against these diseases in the UK, so control is based upon avoidance of tick-infested pastures or through use of synthetic insecticides prior to exposure. Synthetic insecticides last for only a few weeks, so they'll need to be reapplied throughout the tick season. But all of these diseases give some degree of acquired immunity, so it's important in tick-infested areas that there is some exposure to allow the development of immunity. Tick survival is highly dependent upon climatic conditions. So Ixodes ricinus is very prone to drying out, so it requires habitats with high humidities. In the UK, it's most abundant in woodland and upland habitats for this reason, because these habitats contain thick vegetation layers which allow humid microclimates. So upland habitats are characterized by rough grazing areas, and they contain things such as bracken and heather. And this is in contrast to lowland habitats, which tend to be characterized more commonly by improved grassland, short grass with compacted soil. And so in these habitats, ticks tend to be more limited to hedgerows and woodland. And then they're also dependent upon temperature in order to quest. So they require temperatures of around 7 to 10 degrees in order to begin questing. So therefore in the UK, tick activity is seasonal. It tends to range from February to October, with a peak in late spring and early summer. Because Ixodes ricinus is highly dependent on climatic conditions for its survival, it's expected that as the climate changes, its distribution will also change. And there's evidence of distribution change of ticks across different countries in Europe. And because it's highly dependent on specific climatic conditions, it's an ideal candidate for use in predictive distribution modelling. And this can be used to predict the risk of disease to livestock as the climate changes in the future. But to do this, you need data on the current distribution of ticks on livestock farms. So the aims of this study were to compare the relative risk of tick presence on livestock farms in the UK, to use this to identify areas of high risk, and then to use this data to model how this risk might change with the changing climate. In order to collect large-scale data on tick presence and absence across the country, 7,200 questionnaires were sent out to livestock farmers asking about the presence or the absence of ticks on their livestock in the previous year. (926) farmers responded, and here you can see the distribution of respondents. The distribution is similar to the distribution of the underlying farm population, and the respondents were also representative in terms of the ratio of upland to lowland farms and the ratio of sheep to cattle farms. So 13% of sheep farms and 6% of cattle farms reported seeing ticks on their livestock in the previous year. And these maps here showing farms which reported ticks with the red dots and farms which didn't report ticks with the black dots. So it looks like there are areas where there are clusters of farms reporting ticks, but because this is just the raw data, it can't be used to infer areas of high risk, because these clustered areas might also be areas that had a high number of respondent farms. So in order to get past this issue, I looked at relative risk. And these are the relative risk maps here. They're produced using the ratio of the density of farm presence farms to absence farms. So they control for the underlying distribution of the data points. And here, lighter colours are showing increased risk. So you can see that risk generally increases to the west. And then spatial scan statistics we used to test for significant clustering of farms with ticks, to see if they were more clustered than the underlying farm population. And these clustered areas are shown by these bold contour lines here and here. So significantly high risk areas included North Wales, Northwest England, and West Scotland. And in these regions, prevalence of ticks range from 48 to 100%. So that's considerably higher than the averages. These maps were produced in RStudio using the SPAR package, and they use a technique called kernel density estimation to smooth the point data across the whole of the country. But in order to get a finer scale assessment of risk, I built a predictive model to predict risk across the whole country. Oh, sorry, I forgot to mention this. Farms with ticks were significantly associated with upland regions, larger flock sizes, region, and the presence of sheep on cattle farms. So that built a predictive model, and this was a spatial model. It used raster layers, and raster layers are essentially gridded maps at particular spatial resolution, in this case 2 kilometers squared, and they store data in each grid square. And I used 28 of these predictor variables relating to climate, habitat, and host density. And these were used to build a model using the raw presence-absence tick data. So the climate variables I used were bioclim variables, and these are 19 variables relating to altitude, temperature, and precipitation. and they capture things such as seasonality, extremes, and annual trends. So they're biologically relevant. The habitat variables are used related to the percentage of upland and lowland, percentage of woodland and woodland boundary, and the normalized difference vegetation index, NDVI, which is a remote sensing imaging technique which describes the amount of green vegetation. And this has been found to be an important predictor of tick abundance. And then the host variables I used were sheep and cattle density and the probability of deer presence. So then the model I used was a random forest model. And this is a type of supervised machine learning, which uses a bunch of decision trees to classify a binary outcome. So here's an extremely simplified example of a decision tree. In this case, the binary outcome is the presence or the absence of ticks. And then the variables are the 28 predictor variables relating to climate, habitat, and host density. And these variables are used to split the data at each node, so here and here, into yes or no. And then the tree will carry on until all 28 variables have been used for data splitting. But then in a random forest model, lots of these decision trees are built, and the variables chosen at each node are random. So each of these trees will be different, and they will each vote on the outcome at each grid coordinate. And the outcome with the most votes will be the one that's predicted. So because numerous uncorrelated decision trees are used, random forest models aren't prone to overfitting. And another advantage of random forest is that you don't have to specify interaction terms, unlike in linear models. So this random forest model was used with the raw data, the tick presence or absence data, and the 28 predictor variables to predict the probability of ticks being present on farms across the whole of the country. And this is what the model predicted for using current climate data. This map shows the predicted probability of ticks on a livestock farm in any one of these 2-kilometer grid squares, with green showing increased probability. As you can see, the general trends are similar to that produced from the raw data. I tested for accuracy of this model, and accuracy was 93%. And sensitivity was lower than specificity. So this means that sometimes the model wasn't predicting ticks when it should have been. It was giving some false negatives. And this means that the model was probably under-predicting the presence of ticks. So then I used several different future climate predictions using different future climate models to run the model again to make predictions of tick probability under different climate change scenarios. I used 3 different climate models, GCMA, GCMB, and GCMC. And I used a best and a worst case CO2 emission scenario for each. And I did this for the years 2021 to 2040 and 2041 to 2060. As you can see, the predictions from all of the different scenarios were very similar. There were just slight changes in the scale of probability in different areas. So here on the left, you can see the current prediction, and then in the center, an example of a future prediction. And then on the right, this map shows the difference between the future and the current prediction, with red showing an increase in probability of ticks and blue showing a decrease in probability of ticks. So the probability of ticks generally increased across Britain under future climate scenarios, but this relationship wasn't the same across the whole country. And this is probably due to interaction effects between variables. So, for example, winters are expected to be milder in the UK in the future, and this will likely increase tick survival across the country. But this effect is likely to be higher at higher altitudes and higher latitudes, so further north, because in these areas it's currently cold winter temperatures which are limiting tick survival. And then conversely, lower latitudes further south and lower altitudes, it's currently saturation deficit during the summer that's limiting tick survival. So as summers are expected to become hotter and drier in the future, especially in the southwest, this might actually even reduce tick survival in these areas. So I tested for interaction effects with altitude and with northing, which is the equivalent of latitude. And these graphs show predicted probability of ticks on the y-axis and altitude and northing on the x-axis. And you can see that, and these lines here, these grey ones, are predictions from the current climate model in grey, and then the averaged future climate models in orange and blue. And you can see that as altitude and northing increase, ticks are more likely to be predicted by the future climate models relative to the current climate model. So this is similar to findings in other European countries, that as the climate changes, ticks are spreading to higher latitudes and altitudes as conditions in these marginal habitats become more favourable. So in summary, tick infection risk to livestock was spatially aggregated. In high-risk areas, prevalence of tick presence ranged from 48 to 100%. And tick infection risk increased under future climate scenarios, particularly further north and at higher altitudes. So this might further increase risk of tick-borne disease on upland farms. But because climate isn't the only factor that affects tick distribution, future work will involve changing host densities and habitat variables to see how this will impact future risk. And I'd just like to thank my supervisor, Richard Wall, and Hannah for her help with the initial spatial analysis. Thank you.
Thank you, Dr. Katie, for your nice presentation, and we are back to you later. And the next speaker in this presentation section is Professor Richard Wall from the University of Bristol, UK. So are you there, Professor Richard?
I'm here. I'm just trying to find out how to share my screen. Okay.
Please take your time.
Yeah. Okay, is that working? Can you hear me?
Yes.
Good. Thank
Clearly.
you. Okay, good. So I'm going to talk about the use of alternatives to insecticides for managing ticks on pets and on humans. When we started this work many years ago, the first thing we did was a major survey of ticks on dogs in the UK. We did this by recruiting veterinary practices around the country, and we asked those vet practices to collect ticks for us. Over the course of the 3 months of the study, those 1,000 vet practices inspected over 12,000 dogs, and they sent us about 6,500 ticks. It was a very large survey. We found that we had a prevalence of ticks on dogs of about 30%, so that's quite high. We suspect there might have been some over-reporting by the vets, but nevertheless it shows that ticks on dogs are quite widespread and common in the UK. We have 4 species that we commonly find on dogs, and we've heard about Ixodes ricinus from Katie, and this shows the distribution of Ixodes ricinus, extremely common. It's almost 90% of the ticks we find on dogs are this species. We heard about Dermacenta reticulatus from Bryony. This is our least common tick on dogs, and as Bryony said, it's located in some very restricted habitats around the country. But we also find 2 other species, the British dog tick Ixodes canis, and just under 1% of ticks on dogs with this species. And we find Ixodes hexagonus, the hedgehog tick, and that's about 10% of the ticks we find on dogs. It tends to have a slightly more southerly distribution than Ixodes ricinus. Now, there are the reason why we're concerned, as we've heard from the previous speakers, is there are lots of pathogens that are transmitted by these ticks. So we're quite concerned about the presence of ticks on dogs, on pets in general. Now, there are many synthetic pesticides available for tick and flea control. There's a wide range of easily applied and very effective products, but there's growing concern about some of these products. Recently, there's been a paper published in the UK showing that some of the products, the flea products in particular, were being found contaminating waterways in the UK. So there's a lot of concern at the moment about the overuse of these chemicals and whether they cause environmental contamination. So there's growing concern and a desire to try and reduce the reliance on these chemicals in the country. So one of the things that we're particularly interested in is whether we can use plant essential oils for tick control. And we know that many of these plants have active compounds, particularly terpenes and terpenoids, that show insecticidal control. And our work on ticks has shown that they are effective, they can result in mortality, and we think that there are 2 modes of action. First of all, if you apply the essential oil directly to the tick, you get a mechanical effect, and this might be a blockage of the spiracles or the stigmata on the ticks. So there's direct effects of the essential oils. We also know that there's a vapor effect. So if we just expose the ticks to the vapor of essential oils, it will kill the ticks. And that shows that there's some neuroinhibitory effect of these essential oil vapors. We think that many of them will also act as repellents. So when we started work looking at the effects of essential oils on ticks, the first thing we did was develop a simple laboratory repellents bioassay. And this was very, very simple. It consists of a strip of filter paper. The tick is placed in the center of the paper, and a drop of the essential oil is placed at the tip of the filter paper. Above the filter paper, there's another piece of filter paper dosed with an attractant. And in this survey, we extracted the waxes from the hair of dogs, and those waxes are placed on the top filter paper here. The tick is placed in the starting point, and the tick will normally attempt to climb up the filter paper towards the essential oil, and is attracted by the attractant in this filter paper at the top. So we can measure the climbing behavior of the ticks. In some cases, if the essential oil is repellent and more repellent than the attractant, the tick will actually climb down, and in many cases the ticks will also drop off the filter papers. Now, when designing these sorts of simple laboratory bioassays, one of the things I wanted to stress is the very important use of appropriate controls. For the studies that we did, we used 2 types of controls, positive controls and negative controls. Excuse me. The positive controls are compounds that we expect to be repellent to the ticks. And we used PMD, which is oil of lemon eucalyptus, and we used DEET. DEET is the commonest repellent used particularly for biting insect repellents. So these 2 we expected to be repellent to the ticks. And we used 2 negative controls, in this case olive oil, which we wouldn't expect to have any effect on the ticks. And we also used ethanol, because ethanol was the solvent that we used for diluting the essential oils that we used in our experiments. So the using appropriate controls is extremely important in this sort of work. So first of all, using our simple assay, and we screened a wide variety of essential oils. You can see them listed on the bottom of the graph here. And this shows the percentage of the ticks that actually reached the tip of that filter paper. So they climbed all the way up. And you can see with ethanol and essential oils, about 80% of the ticks climbed all the way to the top. We also had good climbing with soya, orange, grapefruit, a whole range of oils. But when we got to our DEET and PMD, which were our positive controls, we got no climbing to the tip. But also we found that ginger, geranium, thyme, turmeric and spearmint were extremely repellent. So none of the ticks climbed to the tip of the filter paper. And we were using 5% solutions of most of the oils and 20% dilution of the DEET. So that's the climbing behavior, but also we found that many of the ticks would just drop off. And when we use our negative controls, our ethanol and olive oil, very, very few of the ticks dropped off that filter paper. But when we use some of the things that we knew were repellent, ginger, spearmint, thyme, a lot of the ticks, about 60 to 70%, after a period of a few minutes, would just drop off that filter paper. So that showed that the essential oils were in fact repellent to the ticks, and it caused them to drop off the filter paper that they were climbing on. So this simple laboratory bioassay gave us a good means of screening different essential oils and beginning to look at their impact on the behavior of the ticks. We then moved on to the second stage, where we went out to the field. Now, as we've already heard from Bryony, the way we collect ticks is just simply by dragging a blanket across the grass, and the ticks will attach to the underside of that blanket. So in this part of the trial, we simply dosed up the cloths, the blankets that we used for sampling, with a standardized concentration. And in this case, we used orange oil, which we knew was not going to, where's orange oil? I can't see it at the moment. I can't see the orange oil. Fourth one, there, orange oil. So we knew that orange oil wasn't repellent, so that was a control. And we used turmeric oil because we knew that turmeric oil was repellent in the laboratory bioassay. And we also used DEET. So we dosed the blankets with these tick concentrations. In this case, we used 2.5% of orange oil or turmeric and 20% DEET. And then we dragged the cloths for a standardized 10-meter walk and then turned them over and counted the number of ticks that are attached. We then held the cloths up and watched and counted and recorded the time taken for the ticks to drop off. And what we found was this is the numbers of ticks on a log scale, the log number of ticks attaching to the cloths. This is the excipient only, ethanol. This was orange oil, where we didn't expect to have any impact. This was turmeric oil, and this was DEET. So turmeric oil and DEET were significantly more repellent than the orange oil or the ethanol only. So you're getting very good repellence effects of these oils on blankets. And this was the tick persistence, the number of ticks that dropped off those cloths over the 1 minute time period. This is the excipient and the orange, so very few ticks dropped off. And with the turmeric and the DEET, almost all the ticks dropped off relatively quickly. So again, showing that these oils and the DEET are repellent to the ticks on the cloths. Well, the final aspect of this particular piece of work was we wanted to know whether the oils actually worked on dogs. So we did a fairly extensive study where we recruited dog owners, and to be included in the study, the dogs that were involved had to have had at least one previous tick infestation. And we had a particular park in Bristol that we were using, and we needed to know that the dogs were walked at that study site at least 3 times per week over the course of the trial. And owners who said that they didn't know whether their dog had had a tick weren't actually included in the trial. And all the owners that were included had to fill in a questionnaire. So they told us about the breed of dog and the age, its sex, whether it was neutered, how often it was walked. So we collected lots of background information about the dogs involved in the study. And for this study, in the end, we recruited 90 dogs, and those dogs were divided into 3 groups. One group was treated with turmeric oil, which we thought would be repellent. One group was treated with orange oil that we thought wouldn't be repellent, and one was an untreated control group. And each dog owner was given a pack, and you can see the pack here. And in the pack, they had a diary, and they had to record how often they walked. And at the end of each walk, they would inspect their dog for ticks, and we gave them a little tick hook here so they could pull the tick off the dog, and they recorded whether they found a tick or not in their tick diary. And they were given a can of oil, a spray. Now, we didn't tell the owners which oil they were getting. Now, most owners probably could tell, because the difference between the smell of orange oil and turmeric oil is quite marked. But just as part of the design of the study, we just numbered the oils A or B. We didn't tell the owners. And we asked the owners to spray the underside of the dog, the belly and the legs, with the oil before each walk, and then to inspect the dog when they returned. And what we found was, this is the control. So this is a percentage of dogs that picked up at least one tick on the belly and the legs over the course of the 4-week trial. So about 70% of the dogs picked up at least one tick. With orange oil, slightly higher, but not significantly higher than the control. But with turmeric oil, significantly fewer dogs picked up a tick over the course of the 4-week trial. So the study showed quite clearly that the use of these sorts of oils could have a protective effect for dogs walked in this particular habitat. Now, one of the problems with turmeric oil is that it's slightly coloured, so it will stain a white dog, for example. So there are various other oils. We found that some of the other oils, like thyme, are also extremely effective. So we could replace turmeric for thyme oil. Now, the most recent development of this work that we've been carrying out was to consider whether we could do similar things in human clothing. Could we use essential oils to protect humans from ticks? And again, we did a field survey. And essentially what we did for this is, these are some of my students, and they made trouser legs out of unbleached cotton cloth. They pulled those trouser legs on, and each of the trouser legs was treated with 5% essential oil, and that was applied on the day of sampling. And the oils they used in this study were thyme oil, spearmint oil, rosemary oil, oregano oil. They also used DEET at 20%, and of course the ethanol solvent control. And this work took place in February and March 2019, and the students pulled their treated trouser legs on and did standard 10-meter walks through the vegetation. And at the end of their 10-meter walk, they'd stand on a little stool, and the other student would count the ticks that are accumulated on the trouser legs, and also they'd measure the rate at which the ticks dropped off the cloth. So they did recordings at 0 minutes and 3 minutes to look at the rate of detachment. And what they found, this is the number of ticks accumulating on the trouser legs, and this is for DEET, so very few ticks accumulated when they use DEET. This is the control, ethanol, oregano, rosemary, spearmint, and thyme. So oregano, spearmint, and thyme were not significantly different from the DEET. So these three oils showed good ability to repel ticks from attaching. Rosemary had little or no effect, and ethanol, of course, was the control. And in terms of the persistence of the ticks on the cloth, as we saw with the other aspects of the study, this is the percentage of ticks dropping off the trousers. This is DEET, this is spearmint and thyme. So with DEET, spearmint and thyme, quite a high rate of detachment of those ticks, significantly higher rates of detachment than we saw with oregano or rosemary or the ethanol control. So the conclusions of these various studies is that essential oils can effectively prevent tick attachment for both dogs and humans. But what we now need to do is further work. We need more extensive field trials. We need to standardize the components of the various oils that we use. And of course, it's very important to have standardized extraction of these oils. One of the things I'm very keen to stress in these trials is that we really need good experimental design with appropriate controls when the studies are undertaken. It would be quite useful to know more about the toxicology of the essential oils, particularly for mammals. We need better excipients, particularly because we need to try and prolong the persistence of these oils. One of the problems with them is that they have very short periods of residual activity because they're quite volatile. And so using better excipients to prolong the residual activity would be very useful for the future. So finally, I just need to thank the 3 students who helped with this work, Penny Good, Ollie Suter, and Freya Cohen. And that brings my talk to a conclusion.
Thank you, Professor Richard, for your nice presentation. And I think that we have to move to the QA discussion for the first presentation section. So we got here many questions to Dr. Bryony, Sen, Dr. Katie, and Professor Richard. So the first question from Dr. Takano from the Japan to Dr. Bryony. Did you investigate field mice during the July to October?
We didn't do any trapping of small mammals. Recently, some studies in Europe have done that. They found that voles seem to have the most larvae and nymphs, but many small mammals did, including mice and other rodents. And they found that the larvae were most active in June, and then nymphs in July to August.
I see. The second question from the Dr. Aneta to Dr. Bryony. So with the reference to the Exodus recurrence, snow cover seems to be important for its survival at the sub-zero. So do you think the same is true for the Demacenta reticulatus?
It's hard to say from doing studies in the UK because we obviously don't get a great deal of snow cover. But as we've seen, D. reticulatus is more cold tolerant. So, I mean, it depends how low the temperature is, but they did survive for 13 weeks at -4. So I expect that if it was much colder than that, it might do, but I can't really say for sure based on UK work. But D. reticulatus do seem to be much more tolerant than Exodus ricinus.
So for the optimal condition, for how long they can survive without blood meal feeding?
Sorry, say that again.
How long, at the optimal condition, how long this species can survive, without blood meal feeding?
So in our studies, the longest was 13 weeks, but I haven't done work specifically looking at how long they can last without feeding. I'm not sure.
Okay,
Richard,
thank you.
Richard's saying about 9 months.
Okay, thank you. And that the other question for you is that from the Dr. Thuy from the NIVR Vietnam, apart from the temperature and the humidity, is there any climate factor can affect the viability of the tick?
I expect sunlight. And when we were sampling them, we also found when wind speed was high, we seemed to collect fewer, but and when it was very sunny. I mean, but that's obviously correlated to temperature and humidity, so we couldn't really disentangle the two. Yeah, and wind speed, we did include that in the model and it didn't come out as significant. But yeah, those would be two that I would expect to.
Okay, thank you, Dr. Bryony. And we have the other question to Dr. Katie. So from the Dr. Tang, for farms with clustered number of ticks, what are the statistics of the outbreak in the area?
So the clustered areas had about a prevalence of 48 to 100% of farms had ticks, and that was a lot higher than the averages, which are I think 6%, around 6%. And there are 3 different clusters. One was in North Wales, Northwest England and West Scotland, and they all had slightly different prevalences. I think West Scotland was the highest, and that was basically all farms were reporting ticks on their livestock. So it did vary a little bit.
And do you think the distribution of the ticks in the farm is depending on the original distribution of the ticks? In some countries, the tick distribution may change because of the artificial movement of the domestic calf and lamb. This is a question for you.
Yeah, so the distribution on farms will depend on the distribution of ticks. So Ixodes ricinus is very widespread across the country. And it will also depend on the specific habitats that the livestock are grazing in. So if they're grazing in habitats with suitable microclimates, then they're likely to pick up ticks. And yes, so what I was looking at was just their distribution on livestock, but not their distribution, not the species distribution. So Ixodes ricinus will be way more widespread than my map showed, because mine were just showing the risk to livestock, and that will depend on both the distribution of the ticks and the distribution of the livestock hosts. And yes, so yeah, tick distribution will depend on the host. So if density of livestock dramatically change, that could change the density of ticks in that region.
And very small question from me to you is that I'm so curious about the data collection process. So who was the respondent in your survey study? This is the veterinary staff, or I mean the livestock owners?
Yes, this was the livestock owners, the farmers. The questionnaires were sent to a randomized selection of farmers, and it was stratified by 6 UK regions. So different numbers were sent to each region depending on the number of farms in each region. And so then it was the farmers that could say whether they saw ticks on their livestock in the previous year.
Okay, thank you so much, Dr. Katie. So here we have some questions to Professor Richard. So how does the dog respond to being treated with the essential oils?
The dogs, as far as we were aware, responded very well. We didn't get any reports of excessive grooming or any other problems from the owners. So we had no reports of any problems at all.
So how about the skin, I mean the irritation? Any report about that?
No, none at all. I think if you use very high concentrations of essential oils, you can sometimes get skin irritation, but we were using a 5% solution. And I think that's pretty safe. We've never had any reports of any response to a 5% solution.
Okay. And how long does it take to take effect on the dog?
It takes effect very quickly. So it takes effect almost immediately. As soon as the oil is placed on the skin, it'll start being repellent. It's not the speed of start which is the problem, it's how long it lasts, because it only actually lasts on the dog for about 8 to 9 hours, because it's very volatile, so the oil evaporates very quickly. So it'll last for 1 day. So it would need to be reapplied every time the dog went for a walk, basically.
And which stage of the 3 hot stick did you collect from the white cloth?
So that again, which species of tick?
Here maybe 3 hot sticks species.
Yes, 3 host ticks. So it's everything we collected on the dogs in our studies was Ixodes ricinus, a 3 host tick.
I see. So as you know that in Vietnam and in many countries, as I know that people are still using the chemical product. I mean that like the pesticide, but actually this is that the big concern about the chemical, I mean the resistance and also the contamination that you mentioned in your presentation.
Yeah.
However, I just curious about from economic point of view. What do you think about the potential use of the essential oils?
Yeah, I mean, it depends very much. If you buy, as we do, pure essential oils, they can be quite expensive. But it can be scaled up. If you use local plants, if there are plants in Vietnam which have insecticidal properties, and there are various examples of community workshops being set up for farmers to extract those essential oils themselves, there's an initial investment in the extraction equipment, but there are ways of making it less expensive, I think. Yeah. But synthetic chemicals at the moment are certainly much cheaper unless you cost in the environmental damage.
Yeah, okay, I see. So thank you so much, Dr. Katie, Dr. Bryony, and Professor Richard. I think time is limited, so we have to move to the second presentation section. We still have some questions, but I think that we can give the audience the answer later. Thank you so much. So right now, I think we should move to the second presentation section. start with Dr. Tawin in Phanket from the Kansas University, Thailand. So Dr. Tawin, are you there?
Hello.
Okay. Hello, Dr. Tawin. So
Yes.
please take your time.
Let me share my screen. Hello. Hi, good evening. Good afternoon, everyone. Today I would like to update to you on the companion vector-borne pathogen in dog and cat from Thailand. And today my talk will be focusing on our recent 4 publication that we have been published this year. First, I would like to give you some background on Thailand. What is make Thailand become a paradise or hotspot for tick-borne diseases? I think from this we can say that it's because of the location, the climate, or the animal as a stray animal population, tick infestation, and maybe this will be lead to the tick-borne infection risk to either animal or humans. From this map, you can see that Thailand are located in Southeast Asia. And as you may know that we are border to Myanmar, Laos, Cambodia, and on the southern part, we are actually a bordering country with Malaysia. So you can see from this, it means that we share the border and it might be at risk of the infected or transmission between endoparasite or ectoparasite between this region. And as you may know that Thailand and other neighboring country, we are located in the tropical zone. So it's will be a lot of chances that this can be a problem. Move to the climate change, climate and weather in Thailand. As you can see that due to the inland nature, we can see that Thailand, we can have warm weather with the high humidity. And some month during March to May, we can have the temperature up to 40 or even 42 degrees Celsius. And after May, then we have the rainforest or rainy season or monsoon season up until October to November. Like this time, we have a lot of rain at the moment. As you can see from this picture, again, that during the rainy season, we also have a lot of rains, but however, it might be different from the UK or from the other part, since it's not gonna be raining whole day. Most of the time, they might rain in the evening or sometime early morning, but during the day, it's only happens sometime that they might have some raining. However, like I told you before, that most of the time is depend on the humidity. It's very high humidity in Thailand. Move to the next problem that we are facing continuously is the stray animal populations. From the livestock department data, we found that actually now we have more than like 6 million dog populations in Thailand, and about 10% of them are stray animal. And even though now we are facing the pandemic of COVID-19, and the problem that are increasing is that some of the household, they have some economic problem, and they cannot actually raise their animal anymore. And again, like you know that we are Buddhist country, Buddhism, and we believe that dog and cat is not, I mean that we have the belief that we cannot kill them. So most of the time that if they are unwanted animal, they will try to leave them or abandon them outside, or sometimes they believe that if they leave them at the temple, the monk or the nun might be a good chance to take care of them. So I think that from the prediction time that we think we might have some more problem or some more abandoned stray animal in the future after this pandemic. And again, that after we have some data on the tick infestation, we found that most of the tick that they have been infesting in our stray animal are Rhizocephalus sanguineus or bowdog tick. From our survey that we have been done, we found that about 96% of the dog population that we have been sampling, they have the evidence that they are infected with this bowdog tick mostly. And however, even though we have a lot of campaigning on the restaining or crystallization campaign, both from the government and both from the university, still that the population of stray animals still rising. And this is maybe still a chance that tick-borne pathogen still circulate or have any problem in the population of the animal. And again, that you can see that most of the time when we go to the temple, we can see that monk, nun, or caretakers, they are very close contact to animal. So in this case, that we can see that this is the one of the factor that they can become a risk to get infected with tick-borne pathogen or any other pathogen that can transmit from animal to humans as zoonotic agents. So we're moving to the first study that we have been done and already published in one of the papers. Firstly, we actually sampling in the stray dog in Bangkok area. And these all dogs are actually living in the temple or they abandoned in the temple, and they have been taken care by monk, nun, or the caretakers. The sample size that we have been collected is we collecting about 360 dog samples. And then we screening about 85 tick sample individually that we collecting from this stray dog. And 5 pathogens that we have been screening by PCR. The first one is anaplasma, second one is Ehrlichia, and then we screen for Barbasia and hepatozoon, and the last one we screen for mycoplasma. And this is the result that we have been found in this study. As you can see that the total infection in the dog, we found about 76%. That is still quite high in this stray animal. And then in the tick, we found about 39% that have been infected by at least one pathogen. And when we look at each pathogen, we found that tick have been infected with around 22% by anaplasma and around 14% by in dogs. While Ehrlichia have been infected more in the dog with 38% and only 3% by tick. And then for Barbasia, we found that 18% are infecting in dogs and 9% in the tick. And for Mycoplasma, we found that 34% infected in dog and 19% in the tick. And the last one, we found that around 20% have been infected by hepatozoon in dogs, and then about 6% in tick. We also investigate about mixed infection or concurrent infections, and we have been found that a lot of the tick, or around 26% of the dog, have been infecting with more than 2 ticks, at least two pathogens within one dog, and then around 8% are infecting with 3 pathogens, and around 2% have been infected with 4 pathogens. Similar to the tick, we found that around 10% are infecting with 2 pathogens, and then around 3.5% infect with 3 pathogens. That is mean that this mixed infection have been going on, and a lot of both tick and dog have been infecting with more than 1 species of pathogen. And next, we're doing some risk factor analysis, and the main association is found that when the tick are presenting in the dog, they have the high risk of infection with any of the tick pathogen in this study. And again, when we do a phylogenetic tree, we found that for Hepatozoon canis and Barbasia, we found that all of the sample are Barbasia vocali. And then Mycoplasma is Mycoplasma himocanis, and then Anaplasma patis and Ehrlichia canis. This is just show you that we have been try to see any SNP that some variable of the pathogen, but mainly they are the same, the pathogen that I have been tell you on the last slide, but we can see that only a few nucleotides that are different from the other reference from the gene bank. The next study, we have been study in the southern part. So we moved from Bangkok to the southern part of Thailand. And why we want to do that? Because of we have collaborated with vet school in from the University of Teng Songkhla from the southern part, and together with also Obihiro University of Veterinary Medicine in Japan. So we actually try to collect also the stray animal from the southern part, and we've been collecting 2 provinces from the southern part. The name is Songkhla province and Narathiwat provinces. And Narathiwat provinces, that these provinces are actually the bordering provinces that are very close to Malaysia. And again, we sampling and screen for the pathogen that called Barbasia, Ehrlichia, Hepatozoon, and Anaplasma. And this is the result that we found that around 6% of the sample are Anaplasma patis, and only 1% are Ehrlichia canis. And then we found around 3.5% Hepatozoon canis. And then interestingly, that we found is Barbasia vocali, but also we found Barbasia gibsoni. Again, with the co-infection, we found that around 4.3% are infected with Anaplasma and Hepatozoon together. And then for around 1% with Anaplasma with Barbasia vocali, and another 1% by Ehrlichia canis with Hepatozoon canis, and around 2.3% with between Hepatozoon canis with Barbasia vocali, and then around 1% that infect with 3 species, Anaplasma patis, Ehrlichia canis, and Hepatozoon canis. Then we interesting for the Barbasia gypsoni because we didn't found any gypsoni before in other part of Thailand. So we screen this with the ITS regions. And when we compare with other study from Taiwan, USA, Japan, Germany, we found that they are really similar to the sequence from other parts. And also interestingly that we are trying to again investigate that the sequences that we have been found is similar to the other part by confirming with 18S. And we found that one of the sequences that is similar to the one from Malaysia as well. So we believe that this might be a chance that this community dog or stray dog, they have might been infected or found the gypsoni species from our neighboring country due to a lot of gypsoni have been reported in Malaysia before. And then Dr. Malaika maybe have some discussion or maybe can talk about that after this. And the next study that we have been done is that we also collecting the same sample in the same community in Bangkok, and we screen for Bartonella. Why Bartonella? Because we want to see if it's the problem of this, even though it might not related to the more on tick-borne pathogen, but maybe on flea-borne pathogen. But then since we also found a lot of flea in dog and cat in this community, so we also study on this. And we're collecting about 808 samples from stray dog and stray cat. And then we screen for Bartonella. And we found that the dog, we didn't found any positive in the dog sample, but we found around 3% in cat samples. And then after we screening by genotyping, we found that the species that we have been found is Bartonella calengii. And then this all of dog and cat sample also have been evaluated for the blood parameter. However, there are no significant difference between the Bartonella positive cats compared to Bartonella negative cats in this study. This is also the same. And then lastly, we are also try to detect Bartonella in tick and flea from the same setting. And again, we collecting the tick and flea from this animal and screen for Bartonella. And we found that in the tick, there have been one tick that have been positive for one Bartonella coenlilase and Bartonella posiensis. But for the flea sample, we found that around 5% have been infected with Bartonella hensale and Bartonella calengii for another 5%. So in this, they have the evidence that they are zoonotic species that they have been found in human as well. So this is just the genotyping or the phylogenetic analysis that show that they are really in the species that I have been tell you before in the last slide. This is the same. We also doing some again that analyze the risk factor that are associated with this pathogen, and we found that the animal that they are living in the outer zone have more chance to infect with this Bartonella compared to the inner zone and intermediate zone. So for the summary of our study, again, that we can say that this vector-borne pathogen still circulate in our community, both in Thailand and southern part of Thailand. And then they also have another study from other group that they also investigate in the northeastern part of Thailand and from the north of Thailand. And they actually found quite similar pathogen, mainly on Ehrlichia anaplasma, Barbasia or Mycoplasma have still been circulate or infect in our animal in Thailand. So as you can see that we cannot actually get rid of this pathogen in our country as yet. And I think the main problem is that, like I told you before, that the climate that is suitable for the ectoparasite to circulate in the environments, and then they can infect with the animals. Finally, I would like to thank my staff, especially my students who are the main investigator of this 4 publication, Mr. Tom Do and Dr. Pirapat Sangsawang. Thank you for your attention.
Thank you so much, Dr. Tawin. We are back to you later. And the next speaker is Dr. Malaika Watanabe from Malaysia. So, Dr. Malaika, are you there? Hello, Dr. Malaika.
Hi, sorry,
Okay.
I was waiting for you to unmute me.
Okay, please take your time.
Sure. Okay, hello, hi, good evening or good afternoon. I'm Malaika Watanabe, and before I begin, I'd like to thank Dr. Duong and Dr. Lin also for having me here today and inviting me to speak. It's a real pleasure, and it's been great listening to the previous speakers. So I actually am from the Department of Companion Animal Medicine and Surgery. So my research interest is infectious diseases, in particular canine tick-borne diseases, but I also am practicing at our University Veterinary Hospital. So I would like to talk about these canine tick-borne diseases from a clinical aspect as well. So as Dr. Tawin mentioned earlier, Malaysia is located within the tropics. And this is Malaysia here, just south to Thailand. And this is the CBD risk map from Bayer. And this risk map looks at the parasite transmitted infectious disease of dogs in Asia Pacific and the risk the dogs in this region have for acquiring vector-borne diseases. So those in red are those countries that are at high risk. And as you can see, Malaysia is red, and therefore dogs in Malaysia are at high risk of being infected with vector-borne diseases. So the important canine tick-borne diseases of dogs in Malaysia are Ehrlichiosis, Babesiosis, Anaplasmosis, and Hepatozoonosis. So I'm going to just touch a little bit about each of these different diseases and then share with you the latest data we have on these tick-borne pathogens. So canine monocytic ehrlichiosis is caused by Ehrlichia canis of the genus Ehrlichia, family Anaplasmataceae. And as you can see, the genus Ehrlichia, there are many different Ehrlichias, but only Ehrlichia canis has been identified in Malaysia as of yet. So we don't have Ehrlichia ewingi, which is also being known to cause disease in dogs. So Ehrlichia canis is a pleomorphic gram-negative intercellular bacteria that forms clusters or morulae in monocytes. And the transmission is via the brown dog tick, Rhipicephalus sanguinis. So this brown dog tick is ubiquitous in Malaysia and in most of the tropics. So this is the main tick that we see on dogs in Malaysia. So for the longest time, we didn't really know how long these ticks, infected ticks, had to actually remain attached to a dog in order to transmit Ehrlichia canis. But a paper came out in 2013, and they identified that the time it actually requires for E. canis to be transmitted is as soon as 8 hours post-application. So this was as opposed to Barbasia transmission, which takes much longer. So what we see in dogs is once they're infected, they can develop different stages of the disease, and the stages are acute, subclinical, and chronic. Obviously, in the acute phases, the clinical signs are quite non-specific. They will present with fever, lethargy, anorexia. And then here in Malaysia, when we see a dog with generalized lymphadenopathy, the major differential is ehrlichiosis, or canine monocytic ehrlichiosis. We do sometimes see dogs presented with hemorrhagic tendencies, and the most common manifestation of that is an epistaxis, as can be seen in this dog. Obviously, in the subclinical stage, they are asymptomatic. However, in the bloods, you are able to pick up a thrombocytopenia. So that's what's consistent in all three stages of the disease. The chronic stage is the same as the acute, but it's more severe, and they often don't respond as readily to treatment. Now, the challenge for veterinarians is really diagnosing or confirming a diagnosis of ehrlichiosis. And the main reason for that is because blood smear evaluation is not very sensitive. So you have only success, in 4% of cases if you actually examine blood smears. And therefore, clinicians also use these point of care test kits looking for antibodies to Ehrlichia canis, but as the name suggests, it's an antibody test kit. So it's a serological test kit, and it only tells us whether the dogs have antibodies. It may be an active infection, or it may have been past exposure. So it's not very helpful, but that's why it's important to make a diagnosis based on a combination of a history of tick infestation, typical clinical signs, and lab findings, most predominantly a thrombocytopenia, and a hyperglobulinemia, in order to make a diagnosis. If you have molecular techniques available, then of course that's the best way to confirm a diagnosis. But often clinicians don't have that method readily available. So next is canine babesiosis, and in Malaysia, there are two different types of canine babesia that have been known to infect dogs. So they are the large form, which is Barbasia vogeli. So for the longest time, we didn't know for sure which one of these babesias. I mean, obviously we suspected it's going to be vogeli because that's what has been found in the region, but we only confirmed that actually recently when my team started doing molecular work and identified these pathogens using PCR, and then obviously going on to do sequencing. So we've confirmed that it's Barbasia vogeli that's found in Malaysia, and the vector is none other than Rhipicephalus sanguinis again. And Barbasia gibsoni has been around for the longest time too. So that was really interesting how Dr. Tawin was talking about how they didn't find Barbasia gibsoni until very recently, and that too in the areas which are bordering Malaysia. So I used to have discussions with my Thai colleagues, and when they said they didn't have Barbasia gibsoni, I said, no, that's not possible. You have it, you just haven't looked for it. So unfortunately, they found it in the area which is bordering Malaysia. So then the question is, did it actually get to Thailand from the Malaysian dogs? So Barbasia gibsoni is the small Barbasia, and the putative tick vector is, again, Rhipicephalus sanguinis. We do know that Barbasia gibsoni can be transmitted via direct blood inoculation when they're fighting dogs, but we don't have fighting dogs in Malaysia. So it's probably transmitted via Rhipicephalus sanguinis in this country. So the clinical findings are associated with obviously the hemolytic anemia that ensues, and therefore the clinical signs include fever, lethargy, anorexia, and then obviously pale mucous membranes due to that anemia. And due to if it is a very especially if it's a Barbasia gypsumii infection, it tends to be more severe. So we do see jaundice and hemoglobinuria or bilirubinuria along with that. And obviously in these dogs, lymphanomegaly and splenomegaly may also be identified. So diagnosing the Barbasias is not that difficult, and it always brings joy to me. Never good news for the dog, but I'm sorry, as a clinical parasitologist, I do find happiness when I actually find the organism, and they are easy to pick up on blood smears. Obviously, though, molecular techniques, PCR, are more sensitive in picking up the organism. Now, the thing with Barbasia is that why it's still the bane of every vet's existence is because, at least for Barbasia gypsumii, it's notoriously difficult to treat. So all the drugs that have been used, we use diminizing acetate, we use azithromycin and atavocone, but as you know, Barbasia gypsumii have developed resistance to them, and therefore there's no drug or protocol as of yet that can completely eliminate infections, and recurrence is very common. So as for canine anaplasmosis and hepatozoonosis, in terms of canine anaplasmosis, the one that is present in Malaysia is Anaplasma platys. We have not been able to find Anaplasma phagocytophila. Obviously, this is probably due to the absence of the tick vector. This has caused confusion for some vets, because the test kit that's available there is a cross reaction between phagocytophila and Anaplasma platys. And for veterinarians in this region who don't know that phagocytophilia has not even been found here, and the one, whenever they do see a positive on the test kit, it's Anaplasma platys. So Anaplasma platys is the cause of cyclic thrombocytopenia, but it's not yet recognized as a cause of major clinical disease on its own. So it is involved in, we have seen a lot of dogs that have been co-infected with one of the other major pathogens. And in those cases, then the thrombocytopenia tends to be more severe and the clinical disease may be more dramatic. But just infection with Anaplasma platys alone, we have not seen severe or clinical disease associated with that. And again, the putative tick vector is Rhipicephalus sanguinis. As for Hepatism Canis, this causes anemia in dogs. Just recently, I was referred a dog that had a hepatozoonosis, and the anemia, the PCV was dropping, and it needed to be treated with imidocarb for, I think, a total of 3 months. So that dog, was eventually recovered, but the treatment duration had to be extended, maybe due to some underlying issues. So there are only anecdotal reports of severe dogs in Malaysia, so I haven't really seen many dogs that actually are presented with clinical disease associated with hepatozoonosis, and it usually is an incidental finding on examination of a blood smear. So the tick vector is Rhipicephalus sanguinis, but the interesting thing with this is that the dog has to actually ingest the tick to become infected, as opposed to the blood feeding for the other diseases. Okay, so just to share with you the work that we've been doing in terms of in determining the prevalence rates of these different pathogens. So this was a study done by my student in 2016, and this was the first comprehensive study that looked at the whole of Malaysia. So a lot of studies, KL, or the capital of Malaysia, lies here, Putrajaya, and the Selangor is the most populated state of Malaysia. So Malaysia is divided into West and East Malaysia. So this is Borneo, which consists of Sabah, Sarawak, and then this is West Malaysia. So this was the first comprehensive study looking at all the different states, and we focused on the roaming dog population. So we sampled a total of 436 dogs, and we looked at, we screened these dogs for the following hemopathogens, so including Hepatism Canis, the Barbasias, Anaplasma platypus, and Ehrlichia canis. So as you can see, this data differs from what they found in Thailand, where we had the highest prevalence rate of Anaplasma platypus, followed by the Barbasias, then it was Ehrlichia canis, and finally Hepatism Canis. So in some of these dogs, we actually found both of the Barbasia. So the dogs had both Barbasia vogeli and Barbasia gibsoni infection. The majority of the dogs that were positive for Barbasia were actually positive for Barbasia gibsoni. So it is a major problem in Malaysia. And we also did notice that a lot of these dogs had co-infections. So 31, almost 32% of them had at least one, were infected with one of the hemopathogens. And then there was a co-infection in a quarter of the dogs had two infections or two pathogens, and then followed by 10% had 3 pathogens. And some of them actually had all 5. Okay, so interestingly enough, though, in the stray dogs, those that were sampled, none of them actually had overt clinical signs of disease, amazingly. So when we look at the concurrent infection patterns, we saw that the most commonly occurring pathogens that occurred together were Barbasia and Anaplasma. And then we had Barbasia and Ehrlichia, and then Anaplasma and Ehrlichia. And so they were, all the different combinations were there. We then went on to see the spatial distribution of tick-borne hemopathogens depending on the area. So we just divided West Malaysia into North, South, East, West, and then we looked at Sabah, Sarawak, and East Malaysia. So as you can see here, the type of pathogen that were most prevalent differed according to the area. So in West Malaysia, you see Anaplasma platypus was the most frequently occurring pathogen. In East Malaysia, it was Ehrlichia canis. In Sarawak, it was Ehrlichia canis. And in Sabah, it was Hepatism canis. So it really differs according to the region. So we focused then on East Malaysia. So we looked at Sabah, Sarawak specifically, and that was because we actually found very high number of infected dogs. So we looked at the serology. We did serological analysis as well as PCR, and here we saw that the majority, the most frequently occurring pathogen was Barbasia, and followed by Ehrlichia and then Anaplasma. So when we look at the serological results, so the prevalence of Ehrlichia canis, so the seroprevalence was higher than the molecular prevalence, and that's what's expected. But what was strange was that the seroprevalence of Anaplasma platypus was actually lower than the molecular prevalence. Now that could just be because these dogs had been acutely infected and may not have seroconverted yet. That could be one of the possibilities. So this is the latest work that we actually carried out, and that was in 2016. So here, this was in 2020. So we actually looked at Peninsula. We focused on Peninsula Malaysia, and these were shelter dogs. So we sampled 220 dogs from 10 shelters, and we looked for Ehrlichia canis, Barbasias, and Anaplasma platypus. So 35% of the dogs were infected with at least one of the 4 pathogens, and Ehrlichia canis was the most prevalent. Again, there were double infection and triple infections that were noted. So in 10% of those dogs, they had a double infection. So the most common was Ehrlichia canis and Anaplasma platypus. And 1% or 2 of the dogs had a triple infection. Then I'd like to share this study with you. So this was a study, a multicentric study. And in this study, we collaborated, the collaborators include Dr. Lin and Dr. Tawin. And this study was looking at the zoonotic vector-borne pathogens and ectoparasites of dogs and cats in Eastern and Southeast Asia. So these were focusing, this study focused on client-owned animals. And these were the these are the locations of sampling. So in various countries, these pet dogs and cats were sampled. And I'd like to share the data with regards to the vector-borne pathogens in dogs. So just to focus on Malaysia here, when they did antibody or seroprevalence, when they checked the seroprevalence, they found that there was 11% seroprevalence of both Ehrlichia canis and Anaplasma platis. But when it came to, interestingly enough, when they did PCR looking for Barbasia gypsini, they didn't find it in any of the dogs. And they did find it in 2.2% of the dogs, Hepatism Canis in 2.2% of the dogs. But that's the difference, you see, because this study looked at client-owned dogs. And the previous studies that I shared with you, we looked at the roaming dog population or the shelter dogs. So the scenario is very different. And this study actually was quite encouraging, because from this study, we'd like to extrapolate that, you know, there's more awareness about ectoparasites and the necessity for ectoparasite control. And thus possibly that is why the tick-borne pathogens that were found, the prevalence was lower. So in conclusion, what we have seen is, I think it's quite obvious from the data I've shared, that there is this huge infection pressure of vector-borne pathogens in Malaysia. And there's an increasing popularity of pets. But along with that, I think we have a very similar scenario to Thailand, where there's a growing roaming dog and cat population. And that, as you've seen, they act as reservoirs of infection and pose a significant risk to naive dogs and cats. So there definitely is a need for greater efforts to curb the tick flea problem for the benefit of animal and human health. But at the same time, we need to think about how we can actually control this roaming dog cat population in order to actually limit the reservoirs of infection. And I think what is important now from the previous speakers too, it becomes obvious with the climate change that the geographic distribution of the tick vectors, and possibly we may even, there may be new tick vectors or other vectors that may be introduced in the region, and therefore constant vigilance and monitoring for emerging and re-emerging tick-borne diseases is essential, especially when it comes to possible zoonotic species that may enter our country. So with that, I'd like to thank you all. And I think especially for those who are listening from the Southeast Asian region or from the tropics, we have updated information at on the website of TROCAP, the Tropical Council for Companion Animal Parasites. And that's always a good reference for veterinarians whenever they're dealing with tick-borne pathogens in the tropics. Thank you very much. And I'll be happy to answer questions towards the end of the session. Thank you.
Thank you so much, Dr. Malaika, for your sharing. Actually, it's very informative presentation. It's very nice to hear you. And we are gonna back to you later in the QA discussion part. And I think we have to move to the next speaker is Dr. Bhui Heinlein from the Vietnam National University of Agriculture. So Dr. Bhui Heinlein, please.
Yeah, I'm here and I will try to share my screen, just a moment. Okay, can you see the screen?
Clearly.
Okay, that's good. Yep. So, good afternoon, everyone. I am very pleased to be here today and sharing with you all the information about tick and tick-borne disease. And I also very glad to listen to all the presentations that you have been done. Very interesting topic, and I could like to share with all of you. First of all, I could like to talk a little bit about our project of strengthening research capacity on tick and tick-borne disease in Southeast Asia, which is supported by UK medications. Sorry, I need to come back. Supported by UK Medical Science networking grant. And I am very proud to introduce with you all of our co-partner, Professor Richard Wong from Bristol University of UK, co-leader project, Dr. Tawin Inpankel from Kasetsart University of Thailand, and Dr. Malaika Watanabe from Putra University of Malaysia. Thank you all for your contribution for the success of workshop and project. And the aim of this project is to engage partner with technical knowledge on prevention of tick and tick-borne disease in order to create a network on research expertise, on exchange knowledge and advanced technique on understanding of tick management. With all sharing from this morning, this own presentation today, we can see very different story between Europe and tropical countries such as Vietnam, Thailand, and Malaysia. So I'm going to introduce a little bit about situation of tick and tick-borne disease in Southeast Asia. At first, we can see that tick is a very common ectoparasite in animals and humans. And besides that, they can transmit a lot of pathogen, dangerous and zoonosis. One of the report of CDC in 2019, they show that about 60,000 persons was infected of tick-borne disease in US. With very high surveillance. And there are about 850 species of tick species used for worldwide. And they can transmit a lot of pathogens such as virus, bacteria, and parasite pathogen. Among of that, virus pathogen is considered as the most diverse group transmitted by the tick. And about 90% of bacteria species can transmit it by tick too. Beside that, many protozoa parasite, as you can hear from other presentation, Professor Dr. Tawin and Malaika already showing that. And Barbasia is considered as an emerged tick-borne disease. So one of the study have reported in 2020, similar same story with Dr. Malaika had been presentation. This study have been done in 9 countries with over 1,000 dogs, and the sample was collected for identified tick in this study, and all tick in this study was recognized as Bicephalus sanguineus. Same study, they found that in 164 tick, which was molecular screening, and see that 22.6% of tick was positive with tick pathogen, including Ehrlichia, Anaplasma, Barbasia, and Hepatism. And among of that, Barbasia was found highest percentage of prevalence. One of study in Vietnam from 2085 up to 2000, 1985 to 1987. This study was collected tick from domestic and wild animal in Vietnam. And the results show that 48 tick species was found in all over the country. One study in 2018 with within 359 dog owners living in around Hanoi, which is in northern province in Vietnam that you can see in the map. And all the tick was found in this study was recognized as Ripicephalus sanguineus 2. And surprisingly, ectoparasite was found in this. Among of that, tick was highest prevalence, up to 76%. And in total of 300.2 tick was screening for tick-borne disease. There are 3 kinds of pathogen was found in this study, including Hepatism, Ehrlichia, and Barbasia. Another study was done in 2015 to 2019 at Bhui province in Kettles. Another kind of tick was found in this study, such as Ripicephalus microplus and Ripicephalus annulatus. Bhui is also one of northern province nearby Hanoi, about 60 kilometers far from Hanoi. And in this study, the prevalence of tick was higher in local yellow cattle compared to milking cows, and which is very interesting point to be discussed later on the control strategies. And the pathogen was found in this study is Anaplasma spp, and very special that in this study, the author found that Anaplasma platis is the first time found in cattle in Vietnam. And in 2020, we have a chance to visit Cuc Phuong National Park, which is located in northern Vietnam, and where located several kind of wildlife rescue center. In that place, we have chance to look for the tick of 27 rescue total, and we found that in both 2 of rescue total species with the high prevalence of tick. And all of the tick in this study found was identified Amblyomma bocardi tick. Randomly collected for 40 tick and analysis by PCR method. The pathogen was found in tick is Rickettsia species. And 75% was found in undone ticks, and the other 20% was found in nymph and larva stage of the tick. One interesting point, when we reviewing 79 pet owners about their knowledge of tick-borne disease from pets, more than 50% of patients will not know about the risk of infection from tick-borne disease from their pet. And more than 70% of them was never prevented tick before. Asking opinion about the tick preventions, only 10% of patients, they agree to have monthly prevention for their pet. And among of that reviews, in conclusion of that review study, even it's very primary study in Vietnam, but we can see that in cattle, farm animals, companion animals such as dog and wildlife in total, we can see different kind of tick and tick-borne disease in those animals, and some of them was recognized as zoonotic disease. So the importance of the risks, how to manage the risks of tick and tick-borne disease in those animals. The story was discussed with farm animals. As you can see, the results in cattle. We can see local cattle with grazing in grass field is more high prevalence of the tick compared to milking cow, which only stay in the cage. And then the educated farmer for tick and tick-borne control is a very important point. Together with regularly diagnosis and treatment tick and tick-borne disease in both animal and environments, highly recommended. The most important point I could like to impress here is the role of policymaker on this, on their role of the tick-borne disease management, because if they understand more about tick and tick-borne risks for animals' health and also human, the control program will be have better policy on that. Similar with pet animals, nowadays in Asia countries such as Vietnam and the other country like Thailand and Malaysia, you can see the similar pathogen and tick species have high prevalence, and also pet animal is increasing rapidly. Together with that, tick and tick-borne control is highly concerned. So educated for pet owner is a priority recommended, together with regularly diagnosis and prevention tick in both animal and environments. And of course, the role of policymaker is highly recommended. Different story with wildlife, because we have very rare, just right beginning of the data screening in wildlife in Vietnam, so we don't have any data yet. And people, of course, not know about the risk of infection, so understand more tick population and tick-borne disease in wildlife. The risk of tick and tick-borne disease notification is a priority recommended, together with the education and prevention tick in environments. And of course, once again, role of policymaker is a very important point in this cycle. So come back to the purpose. Main purpose of this project is building a network on control tick and tick-borne disease. So what I could like to mention here is communication between medical science and veterinary science on One Health concept on tick and tick-borne control. So we need more communication between two parts, and then together have such a network to sharing knowledge, data, and talking about the managed program together with better guideline for everyone. So this is very important and necessary for tick and tick-borne disease control. This is the end of my presentation. Any question and concern, please send to my email, or you can ask me directly. Thank you.
Thank you, Dr. Ling, for your presentation, and we are back to you later. And that the last speaker today is Dr. Nguyễn Văn Dũng from the NIMPE Vietnam, and he's gonna give us some updated information about the situation of tick-borne disease in humans in Vietnam. So, Dr. Dũng, are you there?
Yes, thank you.
Please
I'm
take
here.
your time.
And so, hi, good afternoon, everyone. So now I would like to present my tutorial about situation of ticks and tick-borne disease in human in Vietnam. First, I would like to talk about background. As we know, ticks and mosquito are recognized as the most important vector in transmission of bacteria and viral pathogens to human and animal worldwide. So they can feed on various hosts and transmit or resist pathogen bacteria, protozoa, and virus to from also, most studies have found that Tick-Borne disease in licenses are often limited to specific geographical regions. They may potentially to be found anywhere in the world. Nowadays, international travel from endemic regions to non-endemic regions by people, animal, and cargo can transport this. Tick-bite in Vietnam potential can cause various diseases, include bacteria and viral infection, post infection, fatigue, and unless unless only poorly qualified illness. So exact incidence of tick-borne disease in Vietnam is unknown. So in this review, we will examine Vietnamese tick-borne disease potential of these ticks to carry emerging pathogen of humans and their possible transmission to humans. I talk about Vietnamese tick. There are 896 for this species of the ticks of the worldwide, distributed into main family of Agacidae and Isolidae. In Vietnam, there are 89 species and Vietnam has a unique climate and environment conditions that are favorable for eight of the 14 genus of ticks, and this illustrate key to the Isolidae, Chomiconidae, and Gammasoidae in Vietnam. It's important to note that the ability to carry pathogen in is different from the ability to transmit them, and active transmission has yet to be established in some cases. So in Vietnam, only 5 of 89 human eating ticks act as a competent vector for transmission of pathogen to human. They include Lipicephalus sanguineus, Haemaphysalis spinocerebellis, Haemaphysalis totalis, Amblyopia testudinarium, and Isodes ovatus. So first, I talk about bacterial tick-borne infections, key fever, and some rickettsial infection are the only bacterial disease that's believed to be transmitted by human biting ticks in Vietnam. However, ticks that by human may also be potential vector for transmitting human pathogen that cause anaplasmosis, limb-like disease, and to in Vietnam. This figure about life cycle of Bolena or some other disease in ticks is so that the first stage, adult female drops the eggs. After that, egg hatch into sickless larvae. Then the larvae develop into the limb and infectious limbs feed on human and transmit Bolena. Yes. As the next, the viral tick-borne infection. Alphavirus are a major public health concern in Vietnam, some of which are associated with humidity. The only reportedly placed endemic in Vietnam that's been isolated from human biting ticks. But there are no reportedly viruses endemic in Vietnam that have been isolated from human beating. There are two patients in Vietnam with severe fever with thrombocytopenia syndrome in Hue province in 2017. Which is transmitted by tick, but in 2020, Ningpe has not yet isolated the virus that's called severe fever with thrombocytopenia syndrome from ticks in Hue. And the other tick-borne disease go about Barbasia, tick paralysis. Yes. And the fifth, I saw some recent ticks transmit disease in Vietnam. And the first, some recent tick transmitted disease on patients at hospital in Vietnam from 2017 to 2019, but unpublished data. The reason qPCR for rickettsial fever, typhoid, and qQ's fever. Total patient joined to research 2,583. So these are shows that 67.9% negative by using PCR method, but 32.1% positive equates 79.5% typhus, 11.1 fever transmitted by fleas, 4.8% rickettsial fever, and 4.6 Q fever. Currently, we have map of the distribution of this disease in Vietnam, but not yet published. So we are not allowed to show them here. I'm so sorry. In 2017, in the southwest, the rickettsial pathogen has been identified in Repellent sanguineus with an infection rate 1% out of total of 299 collected ticks from 2015 to 2017. 34.1% of patients infected with typhus, and 3.3% patients infected murine typhus. Among the patients treated at National Hospital for Tropical Disease. In 2020, 38 out of 88 patients are done caused by rickettsial SPP in some hospital in Hanoi. In Military Hospital 103 and Military Central Hospital 108. There is there is not an adequate and systematic study on the roles of ticks and in disease transmission. The previous study just focused on the presence of rickettsia in a human. So I have some conclusions. First, in Vietnamese climate and environmental conditions provide suitable conditions for many ticks. Vietnam has known to be endemic for 89 of the known 896 tick species. At least 5 species in Vietnam takes attacks to and feeds on domestic animal and human. While tick-borne pathogens of humans don't appear to contribute to Vietnam overall communicable disease burden, this perception must be researched to confirm this statement. Again, a true baseline estimate of the burden of illness associated with tick bite we can then prepare for the future when change in the climate, times lifestyle, human and animal population will invariably impact on the livelihood that tick bite will in many parts of Vietnam. So further research according Vietnam undoubtedly faced new disease threats associated with tick bite, all of which can only be countered by improving our knowledge of ticks, the pathogens, and the epidemiology of tick bite and its consequence. They urge needs to be comprehensive to study to determine the roles of Vietnamese human biting ticks in transmission of emerging pathogen to human in Vietnam. These are some here some picture of our activity when researching tick and tick-borne disease in Vietnam. We keep tracking to collect ticks. Recently, we held a training workshop on ectoparasite and Rickettsia pathogen taught by U.S. Navy experts. Yeah. We have a museum of medical etymology storing thousands of ectoparasites collected from 1970s, including ticks. Thank you for your attention.
Thank you, Dr. Dũng, for your presentation. So after hearing the presentation from the Dr. Dũng and the Dr. Linh, I think that we still have a lot of things to do. Anyway, we have to come back to the QA discussion section. So the first question from Mr. Nguyen to Dr. Tawin. Do you have any evidence regarding the infection of the pathogen like the canine tick-borne pathogens in humans in Thailand?
Thank you for the questions. Actually, it's one of our problem as well, since most of the vector-borne pathogen in humans are mainly focusing on rickettsia or Bartonella, or some other mosquito-borne like dengue or malaria. So the one that actually from tick, we don't really have a lot of publication or evidence on that. So that's why I think from this discussion, we still can see that a lot of work that we need to do, especially on One Health approach, that we need to work together between humans, medical side together with veterinarian sides. Yeah.
And we have two questions from the Professor Richard Wang. So which tick life cycle stage did you collect and test? do the animals at the monastery get tested for the disease?
Actually, all of the dog and cat are asymptomatic. So mostly they are fine, but since we don't really know that it's because of they don't get enough food or they get maybe endoparasite as well. So we don't really can say that it's because of vector-borne only, because some of them are having like mixed infection between endoparasite and vector-borne parasite. And back to the first question, that we actually collect both NIM stage and adult stage. We try to not screen by both stage, but it's depend on which state that we collecting from that dog. So yeah,
I see.
answer that we get both NIM state and adult stage.
Okay, thank you. And the next question for you is that, are there any report, from the monk, at the monastery, that they were infested by any ticks and fleas?
So far, we didn't get any complaint when we collecting the sample. But however, I think still some of them might have some experience, but they don't know about it. You can see that most of the monks are very close, mostly with the dog. And from experience that we went to the temple, we can see that some of them actually really bring the cat. Cat can roam freely in the temple, right. And they also can go into the room of the monk sometimes. But the dog mostly they will stay out. In the area, not really inside the bedroom or the room that the monk staying.
I see. And the last question for you is that how did you avoid the confusion between the pathogen in the blood mean of ticks or it in the tick body?
What do you mean by that? Because when we extract, we extract from the whole tick. But again, we actually wash them first by PBS before we extract. So it means that the pathogen will be from the inside. However, we since some of them might be engorged or maybe they're feeding the blood. So we cannot say that actually it's really from what you can call it from the blood that the tick are sucking from the dog or cat, or or they actually replicate itself inside the tick.
Okay, thank you, Dr. Tawi. And the next question is to Dr. Malaika. So from the Dr. Phương from Vietnam. So he works in the hospital too, and he's one that you share more information about the treatment of the Ocular Canis and the Barbasia infection in dogs. How many % of those cases are curable, and how long the treatment takes?
So actually, with regards to Kiosis, we usually just treat with doxycycline at 10 mg per kg SID, and we usually go on for 28 days. So that's the recommendation. And most of these dogs, they do really well. I think the only problem is sometimes we encounter those that are in the chronic stage, so they don't respond so readily. And some of them, obviously, they have immune-mediated complications, so maybe immune-mediated thrombocytopenia or immune-mediated hemolytic anemia. So in those cases, we usually add on some steroids for a short term, and they do really well. So overall, I think in terms of our cases of acute monocytic ehrlichiosis, the treatment goes on for a month, but these dogs, they show rapid recovery. In just 2-3 days. You see a significant change in, in terms of their eating again, return of appetite, and they're becoming more alert and responsive. As for Barbasia, I think that's always been a challenge. With Barbasia vogeli, it's more straightforward. So we use diminazine acetate. So that's licensed for use here. So we use that, and we usually just give one dose, and that tends to be sufficient. However, with Barbasia gypsoni, we've had a lot of difficulty. And for our hospital, we have a protocol where we start off with diminazine acetate. We give 2 doses, obviously at the lower dose range, so as to avoid toxicity. And then we try, your atavocone azithromycin. It tends to be expensive, so that's why we don't always get a chance to use atavocone, because owners can't afford it. And then finally, when that doesn't work, we would go for the 3-drug protocol. So the 3 different types of antibiotics. There are a couple of different protocols I use, but we've had to use that in some cases, though I really make sure that that's a last resort, because we are just really worried about microbial resistance. So I hope that answers your question.
Okay, and the second question for you from the Dr. Bryony, UK. How common is that dogs may be infected with the Barbasia but not show any clinical sign?
When it comes to Barbasia gypsoni, I think most of the infections that we do see, they tend to be clinical. Obviously, though, there is that percentage which are able to maintain the Barbasia gypsoni levels because of strong immunity. They never really, you don't really get high parasitemia, because even with treatment, as we know, it doesn't necessarily completely eliminate infection. So when they're able to just maintain or to ensure that, they keep that, multiplication at a minimum, they don't show any clinical signs. With Barbasia vogeli, what we noticed that it is one of the least pathogenic. So we usually see this more of a problem in young puppies, and then in those dogs that are immunocompromised or are on some, immunosuppressive therapies. So that's when we see it the problem. So there is that certain population which would be harboring these Barbasias and not be showing clinical signs. And that became even more obvious in our studies, where we actually sampled the roaming dog population that were infected, but they were not showing clinical signs. But obviously these stray and roaming dogs have a more robust immunity compared to our pet dogs.
Yeah, thank you so much for your very clear answer. So that the next question, I think that to the Dr. Ling from Dr. Aneta. So does the infested turtle have any discernible clinical signs, Dr. Ling, in your study?
Thank you. Actually, we didn't see any symptoms or clinical signs from turtle. That's a very big concern, because they have such kind of high prevalence of Rickettsia pathogen, but they don't have any in tick, but they don't have any clinical signs when they have many tick infections.
Right.
So really concerning.
Right. The next question for you is from Mr. Nguyen. Have you ever checked the Barbasia species in the Kettles in Vietnam? Is there any genetic relevance among these pathogens in dogs and cattle?
That's a good question. Actually, in this review or presentation, I didn't mention about Barbasia in cattle, because this is not publication yet in our study. So yes, of course, when we checking in cattle, we saw also Barbasia and another kind of protozoa Theileria as well, but it's not publication yet. So beside of anaplasma found in cattle, we can see another pathogen as well.
And he also wonder about which species of tick are prevalent in Vietnam. I mean, here maybe the cattle tick.
Yeah, actually, we are on the way to identify the species. Yeah. Okay. So we will confirm later by publications.
I see.
Yes.
And the last question for you from the Dr. Phúc from the Vô Hun, Vietnam. Can dogs get the limb disease from the tick bite and have any vaccine prevent it for dogs?
Luckily, up to now, we have no finding with Bolaria in Vietnam, and also the vector isodic ticks in Vietnam. So I don't think we have that such problems. But for tick control, because you can see tick-borne disease was show many pathogen transmitted by tick. So we're thinking of researching more about tick vaccine. So hopefully in the future we will have it. But in the world they have some of the vaccination for tick and some disease as well.
Okay, thank you, Dr. Ling, for your answers. And then I think that we have the two questions to Dr. Dung. Is there any connection? Yes. Yeah, is there any connection the presence of the rickettsia detection in humans and ticks in animals in Vietnam?
Yes, now there are no any connection. The presence of rickettsia SP detection in human and ticks in animal in Vietnam because you know ticks and Tick-Borne disease is as neglected field in Vietnam, so they receive a little research attention. There is not any genetic relevance among these pathogens in human and ticks. Yes, so there is not connection. Yeah.
So you mean that as far as you know that there no, I mean that the publication any report about the connection between.
Yeah. I think in the future I will do it, and now we just use testing your ICG support ICG with rickettsial cell groups. So I cannot identify rickettsia species. Yes.
I see. I understand. One small question from me. Actually, I'm so interested in the two cases positive with the SFTF virus in the Hue that you mentioned in your presentation. So this is that the local, I mean the domestic cases, or this is the imported cases?
Local. But in 2020, me and some colleagues go to Hue to collect ticks and PCR to detect SF but has not to detection. Yeah.
Dr. Go. I think that Dr. Ling want to ask something. So please, Dr. Ling.
Yes, thank you for the question. Regarding to the question of Dr. Dung,
yes.
I actually have some information about the project of rickettsia in humans in medical science. I mean, so at that time, I tried to connect to the tropical hospital. To asking the cooperation between human and animals on this disease, Tick-Borne disease, because I think it's important, and we already found some pathogen in animals. So the most important point is how to communicate between medical science and veterinary science together to fix the problem. So the role of Dr. Phúc from Vô Hun, I think is very important.
Thank you.
So thank you so much, Dr. Dung and Dr. Ling for your sharing. And I think this time's up. So I think that it's time for the graphing that today workshop. So on behalf of the organizing, I mean the team, thank you so much for the attendance and thank you so much for the attention. And please let me remind you that our staff will make that the breakout room in the Zoom platform for the speaker and guests. So for the panel discussion, so don't leave us. And for the other, thank you so much for your attendance and attention. So stay safe, everyone.
Okay.